gated channels that are next to voltage gated channels. So it's a very slow propagation of that action potential. With saltatory conduction, where you have myelination, you can skip large amounts of axon. Large is relative because we're talking about something that's really small. But you can skip underneath the myelin and you don't have to open any voltage-gated channels under the myelin. Just have the charge move and not have it become too much less due to diffusion under the myelin to the next node of Ranvier and open those voltage-gated channels. So when you have a contiguous neuron, it takes a while for the message to get to the endpoint. Now we're talking about maybe 3 to 10 meters per second. But a saltatory conduction neuron can send it 180 meters per second. So it's a big difference. Both have to generate local currents to then get the next bunch of voltage-gated channels open. So in the contiguous neuron, it's voltage-gated channels next to the previously open voltage-gated channels that are being opened. So maybe a couple of channels are opened by current that diffuses downstream. The upstream, there is diffusion upstream, but you're running into the absolute and relative refractory periods of the channels upstream. So there will be no effect of sodium that diffuses backwards. Only downstream will you get the channels open. All you have to do is get enough sodium in to cancel the negative charges that are right at the plasma membrane. Positive charge leaving the outside automatically leaves negative charge behind. So just by you're canceling the negative charges out there, and now you've got fewer positive charges, there's going to be more negative charges available. So this is going to take time for each little segment, each little chunk of 2 voltage-gated channels to open along the length of the axon. You can increase the speed of conduction in unmyelinated nerves, try and say that fast when you're tired, by increasing the size of the axon. But you run into normal space, so you can't increase the size of the axon anymore. So create a myelinated axon. You can have a small axon and have it conduct about 10 times as fast as an unmyelinated axon. That's huge. So creating local potentials, you have to reach threshold downstream, open those voltage-gated channels. You get an identical action potential at those channels that you had upstream. So it's called regeneration or propagation of the action potential. So you reach threshold, you get an action potential as the as the sodium is coming in and diffusing downstream. The next segment is reaching threshold. Nothing's happening too far downstream because the sodium dissipates, doesn't get to threshold, may even totally disappear if you're too far downstream. So here they're showing you at resting membrane potential. So no sodium got that far. Before it diffused away from other sodiums and before the ATPase has pumped it back out. So here we've got a complete action potential, and the next one is starting, the next segment is beginning to start the depolarization of the action potential curve. The next segment is reaching threshold. So you complete one action potential, the next one is taking off in the next, forming in the next segment. This part of the neuron is going into its refractory, relative refractory period. So any sodium that comes in due to these channels opening is not going to be sufficient to get another action potential upstream. Remember, this is going to be below minus 70, it's going to be down around minus 80. So no effect of the diffusion upstream, but downstream you're getting to threshold. And as this one then completes the repolarization phase, this one will be taking off in the depolarization phase. So each successive little chunk of the axon has to regenerate the action potential by opening the voltage gated channels.
An action potential occurs at the first node. Sodium ions flow into the axon through sodium channels in the node. Local current flow depolarizes an adjacent node. The second node reaches threshold and an action potential occurs. Sodium gates open and sodium rushes in. At the same time, an outward flow of potassium restores the first node to resting potential. In this manner, the action potential jumps from node to node and is propagated along the axon.
How many of you understand why the positive charge, when it arrives at the node of Ranvier, has a positive charge on the outside that migrates backwards? Say that a little bit louder.
Because it's a negative now, because the positive is sweeping in, so that positive is going to want to be attracted to the other negative.
Yes. So the positive charge that left left a net negative charge behind. The positive charge isn't just at the nodes of Ranvier, it's all on the outside of all the axon. So the positive charge that leaves to go in here to counteract or to reverse the negative charge inside, it's going to leave a negative charge behind, excess negative charge behind, and the positives that are over the myelin move towards the negative, and this positive moves towards the negative. I guess to just clean it up a little bit, they've removed the intervening positive charges, but this is interstitial fluid. This is going to have an overabundance of positive charge, but mixed in with negative charge. As positive charge disappears and you leave negative charge behind, then positive charge will be attracted to that negative charge.
Dendrites are projections from the surface of a neuron's cell body. Dendrites receive signals from other nerve cells and carry those signals toward the cell body. The cell body is the part of the neuron that houses the nucleus and organelles. The axon, or nerve fiber, is an elongated tubular extension that conducts action potentials away from the cell body. The first portion of the axon is the axon hillock. This is the site where action potentials are triggered by a graded potential if it is of sufficient magnitude. Graded potentials occur in response to input from other nerve cells. The action potential is conducted along the axon to the axon terminals. These terminals release chemical messengers known as neurotransmitters that influence other cells. An action potential is initiated at the axon hillock. Once the action potential is triggered, It is automatically conducted down the length of the axon without the need for further stimulating input. Imagine you are looking at a longitudinal section of the axon. The area at the far left is at the peak of an action potential. Watch as local current flow induces an action potential in the adjacent area. The membrane potential in this area is shown in the graph just above it. Because opposite charges attract, current flows between the active area and the adjacent inactive area both inside and outside of the axon. This brings the inactive area to threshold. At threshold, sodium rushes into the cell, triggering an action potential in the previously inactive area. At the same time, the original active area returns to resting potential because of potassium efflux. Again, local current depolarizes the adjacent inactive area, triggering an action potential here. Notice that the original action potential does not travel along the axon. Instead, it triggers an identical new action potential in the adjacent area of the membrane, with this process being repeated along the axon's length. In this way, an action potential is automatically conducted to the end of the axon.
So conducted, propagated, regenerated, it's being reformed in each segment, either contiguously every single section of the axon or from node of Ranvier to node of Ranvier. The myelin is what is allowing for current to flow without decrement or very little decrement for a particular segment of the axon. This is an insulation. It's a plasma membrane of a cell that is wrapped multiple times around a segment of an axon. In the central nervous system, it's an oligodendrocyte. Oligo means a few. So you have one oligodendrocyte to several segments of an axon, or several segments of several axons. So we'll see this on a later slide. So we have a ratio of one cell, but with multiple foot processes wrapping multiple segments of multiple axons. A Schwann cell is in the peripheral nervous system, and it has a ratio of Schwann to axon segment. It's 1 to 1. So Schwann cells forming myelin. One Schwann cell wraps one segment of one axon. Oligo, one cell wraps multiple segments of multiple axons. Nodes of Ranvier are the bare spaces in between the myelin. And these bare patches are where your voltage-gated sodium and potassium channels are located. So current can flow underneath the myelin to the next node without loss. There's not much space under the myelin for ATPases to pump anything out. Very few ATPases there. No need, really. And also very few sodium channels under that. So voltage-gated sodium and potassium channels. This is going to become a problem when you start to develop a demyelinating disease. When the myelin disappears, now you've got segments of axons that are contiguous again, but no voltage-gated channels under these segments where the myelin has, or in the segments where the myelin has disappeared. So you get a fast propagating action potential, then it hits a region where there are no voltage gated channels, and it pretty much screeches to a stop and has to go section by section again. So myelin, when you see the nucleus of the cell as the external surface of multiple wrappings, this is artist made a boo-boo here. The nucleus is external to the myelin, not internal to the myelin. When you have one Schwann cell to one segment of one axon, that 1-to-1 ratio, this is peripheral nerve myelination. For central nervous system, you've got a cell here that is separated, a cell body is separated from the axon, sends out different processes, and these multiple processes wrap multiple axons. So we've got 3 axons here, and this one cell is wrapping multiple segments of multiple axons. So this is central nervous system. Central nervous system, you don't have the luxury of space. You've got to get myelin created in close quarters. So one cell being able to myelinate several axons, it allows for the myelination to occur with less cell body in the way. So here in the peripheral nervous system, the Schwann cell wraps around the first time, and then the plasma membrane wraps underneath the external original layer, and then wraps again and again and again internally, so that the nucleus ends up being on the outside with respect to the internal layer of myelin. Whether you have the myelin formed by a Schwann cell or you have it formed by a oligodendrocyte, its function is the same. It's insulating the axon. The voltage-gated sodium and potassium channels will be located at the nodes of Ranvier. There will be very little under the myelin. You don't have access to the extracellular fluid under the myelin anyway, so don't put very many voltage-gated channels there. The fact that you have all this fatty plasma membrane wrapping the axon means that there's not much space there for ions to diffuse out. And so you don't lose the charge that's coming in at the node of Ranvier. It's able to go under the myelin, and it's said to jump to the next node. Just moves very quickly without the dissipation of the ATPase and loss of the charge. So here we've got a node of Ranvier. It's got voltage-gated sodium channels, voltage-gated potassium channels. The sodium channels open and let positive charge in and leave net negative charge behind. The sodium then diffuses both directions, upstream and down. Downstream, it can open the next voltage-gated channels downstream, but as it's opening those channels downstream, this node of Ranvier is going into its absolute, and it is in absolute refractory period as soon as the voltage-gated channels open. You absolutely can't open them again while they're already open. That's the definition of absolute refractory period. Once some of them close, they close with the inactivation gate. You still cannot open them again until they get back into their original conformation, which is that inactivation gate open and the activation gate close. This is when you're back to resting membrane potential. So having fewer sodium channels to open and also having potassium channels open and allowing potassium to leak out means that this segment is, once the potassium channels open, you're in the relative refractory period. it's going to require more sodium to get you to threshold than will be supplied by sodium diffusing backwards from node of Rambier downstream. So with myelin, the fast movement under the myelin sheath from node of Rambier to node of Rambier, again referred to as saltatory conduction. Sauter, look it up, from the French, meaning to jump. Little jump, not like a jeté, grand jeté, or anything like that. It's actually a little jump. The propagation is about 50 times, up to 50 times as fast when you have myelin. The local currents still are created. That's what gets you to threshold. And then once you reach threshold, the action potential is produced. So here, you open voltage-gated channels upstream, diffusion going downstream and upstream. Always diffuses upstream, it's just random movement of charges. Upstream, you're running into a refractory period. Downstream, you're at resting membrane potential. So you can open the voltage-gated channels down here, and upstream, you're going to need more sodium, and it's not coming from these channels in order to get an action potential upstream. So voltage-gated channels here, open voltage-gated channels in the downstream node. And when enough sodium comes in that you're at plus 30 millivolts, sodium channels close, potassium channels open. So as soon as the sodium channels close, potassium starts to leak out. It's going to make it harder for you to get an action potential in this node. Downstream, sodium's rushing in, and you get current flowing in both directions, but you're in a refractory period upstream, and you're at resting membrane potential downstream, so you get what a regeneration of the action potential at the downstream node. So this shows you that the sodium that entered first node activates the second node. You're at in the relative refractory period now that the potassium channels are open. This makes it really difficult for that sodium to generate another action potential upstream. You saw that. What we're talking about so far, we talked about ligand-gated channels, and those are chemical. So either a hormone arises via the blood, or a neurotransmitter is released and diffuses across a synaptic cleft to the receptor. Electrical synapses, very rare in the nervous system, a lot in the heart, but in the nervous system, they're very rare. There you get the current actually flowing from inside one cell to inside another cell. So gap junctions, remember, they're connexons, series of 6 proteins that create a channel between the cytoplasms of 2 adjacent cells. Whether the current flows through a gap junction or it's created by channels opening because ligand bound to them, you get the same effect. You get a change in the charge inside the cell. So one cell body and its dendrites are probably receiving close to 1,000, if not more, synapses from various neurons. So what you see here, some magenta, some green, some blue colored axons. These are from different neurons. Some will be excitatory, some will be inhibitory. They have to add all the inputs of all the charge. Positive if it's excitatory, negative if it's inhibitory, and you have to get all those charges adding to threshold, which is usually about 20 millivolts in the positive direction, plus 20 millivolts at the axon hillock. Remember, axon hillock, the old term for initial segment and trigger zone. Those give you more information. Trigger zone tells you that's where the action potential came from or is initiated. Trigger zone is triggering the action potential. Axon hillock just tells you it's a little moundy shape of the axon that's closest to the cell body. So over here, actually a scanning electron micrograph showing you a lot of different neurons are going to have their effect on this cell body. Maybe not all at the same time, but you can change the inputs from different neurons. And then sometimes you have some to depolarize, other times they sum to hyperpolarize, and that neuron is less likely to fire. Sometimes they sum to no change in resting membrane potential, and nothing happens to the cell at all. So chemical synapses, we've drawn this many, many times. You've got a receptor in the dendrite, receptor in the cell body, and you're going to have a chemical bind to that receptor and open a channel. The channels may allow positive charge to come in. They may allow positive charge to go out. This is having opposite effects on the inside of the cell. So if you have positive charge come in, you get closer to the threshold. This is going to be called an excitatory or depolarizing potential. EPSP stands for excitatory postsynaptic potential. We're talking about the neuron that is being stimulated by the chemicals. If positive charge leaves the cell, then you have an IPSP, an inhibitory postsynaptic potential. We'll come back to that, but I want to use these terms earlier, so I want you to know what I'm talking about. Okay, so chemical synapses, the neurotransmitter is going to bind to a receptor, but that receptor is going to determine what the effect of the neurotransmitter is. Your body will make fewer neurotransmitters than receptors. Each cell can select what type of receptor it puts in its plasma membrane. The neurons can release the same neurotransmitter, and it binds to two different cells and has opposite effects. So if it opens a sodium channel, the neurotransmitter is depolarizing, it's creating an excitatory postsynaptic potential. But if that neurotransmitter opens a channel and positive charge leaves the postsynaptic cell, it's an inhibitory postsynaptic potential. The statement here that each presynaptic neuron always releases the same single neurotransmitter, we're going to keep this. It's okay for now to believe that, but it's really not true because some neurons release more than one neurotransmitter. And we're going to find out that some of them release polypeptides that are called neuromodulators, and they are coming out of the same presynaptic neuron. So for now, you can think that all neurons release one single neurotransmitter, but be aware of the fact that's not entirely true. Some release more than one. Yes?
What are we getting from crosstalk?
At the end of the chapter. Yeah. So I don't want you to be, what did she just said earlier? It's a single neurotransmitter, and now she's talking about more than one. Yeah.
So on our next midterm, if you ask us on any, is it going to be one or more than one?
By the end of the chapter, it's going to be more than one. Yeah. So most If it said most presynaptic neurons release one neurotransmitter, that would be more of a true statement. But there are quite a few, but not many, who release more than one. And they're finding more that release more than one. So the numbers might even out as they discover these newer scenarios. Okay, action potential is not jumping over, it's jumping under the plasma membrane. When the action potential that is indicated by these arrows arrives at the nerve terminal, it's going to open voltage-gated calcium channels. If you remember your muscle physiology, smooth muscle, cardiac, and skeletal, they all, once calcium gets into the cytoplasm, then contractile filaments start to interact and you start to get shortening of those filaments. These vesicles are attached to the plasma membrane by contractile filaments. When the calcium comes in, the filaments shorten, the vesicles move to the plasma membrane, and then the proteins interact with the plasma membrane. That's called docking. And then there's release of neurotransmitter by some form of exocytosis. What's being shown here is what's called the classical exocytosis, where the vesicle goes into the plasma membrane and then totally regurgitates all the neurotransmitter into the synaptic cleft. There is another type of release which is called kiss and run. The vesicle comes, fuses, but just opens a little pore, and you can think of some saliva coming out while you're kissing somebody, but nothing else is coming out, just a little bit of neurotransmitter, and then the lips close and you run away. So that's somebody's romantic description of this type of vesicular exocytosis. You kiss the membrane open enough to let a little bit of neurotransmitter out, then the vesicle fuses again and pulls away from the plasma membrane. Neurotransmitter diffuses across the cleft and binds to the receptor on the postsynaptic cell. So this postsynaptic neuron is then going to have local potentials created in its cytoplasm. What those local potentials are depends on what channel is open. If you open sodium, you get an excitatory postsynaptic potential. If you open potassium, you get an inhibitory postsynaptic potential. If you open a chloride channel, inhibitory postsynaptic potential. So write this down and think about it. Depolarizing or hyperpolarizing. EPSP, IPSP. Oops, sorry. So increase concentration of sodium inside. That means you open a sodium channel. Increase concentration of potassium inside. That means you closed this channel. This one is open. Decrease concentration of chloride inside. That means you closed a chloride channel. You have to remember you have an ATPase that pumps chloride out. So if you close the channel that lets it come in and pump chloride out, you start gaining positive charge inside the cell. IPSPs, the opposite of EPSPs. How do you get an IPSP? Decrease sodium in. So you must have closed the channel. Decrease potassium in. That means you open the channel. Increase chloride in. That means you open the channel. So EPSPs are opposite from IPSPs. I mean, that's already intuitive, but what are you doing? First, you have to figure out what creates an EPSP. Sodium concentration going up by opening a channel. Sodium's in higher concentration outside. Always, sodium is higher outside than inside any cell in the body. Open the channel, sodium moves in, down its electrical and concentration gradient. You get an excitatory postsynaptic potential. If you close channels that let potassium leak out, then it accumulates inside. Also remember there's an ATPase pumping it back in. So it accumulates positive charge quickly, creates an EPSP. Closing chloride channels, chloride diffusion in is less. There's an ATPase pumping chloride out, so positive charge starts to accumulate due to the ATPase. Slower, but still can get you EPSP. And then IPSP is exactly the opposite. Close the sodium channel, so less sodium gets in. Open more potassium channels, so more potassium goes out and decreases potassium concentration inside the cell. Or open a chloride channel and let negative charges come in. All of those will get you further from resting in the negative direction. All of these will get you closer to threshold. If you open exactly the same numbers, then nothing happens to resting membrane potential. Everything cancels out. So this is just an actual transmission electron micrograph showing you all the little vesicles that are there full of neurotransmitter. The synaptic cleft, it's kind of black in the middle because that's where the neurotransmitter has been released, and more metal has been picked up in that location. So presynaptic neuron, postsynaptic neuron. This animation didn't work, so I'm just going to skip it. But you have to meet, all neurotransmitters have to meet a checklist. I think it's like 7 things long. They have to find a receptor for it. They have to find an enzyme that breaks it down. It has to be released from postsynaptic terminal. I forget, there's all these criteria that have to be met before you reach neurotransmitter status. There are a lot more things that are putative, mean think that they might be neurotransmitters, but they haven't met all the criteria yet. And then we have some that are also, I think it has to be, I think on the checklist it has to be really small, like an amino acid inside. If you look at all of these, they are made from amino acids, acetate stuck to choline, acetylcholine, dopamine, norepinephrine, epinephrine, all made from tyrosine, serotonin from tryptophan, glycine is already glycine, glutamate, aspartate, Those are all amino acids. These are modified. Histidine is going to become histamine, and so forth. And this one is a modified amino acid. These are the neurotransmitters that are most common in the body, but there are others, there are a lot of others that we'll learn as we go through different organ systems. Okay, so we learned about what it takes to get an EPSP. You've got to have positive charge accumulating in the cell to cancel negative charges right at the plasma membrane. So you reverse the charge across the plasma membrane. Get it positive inside, negative outside. Easiest way to do that, let sodium come in, leave less negative, more negative charges outside, or close potassium and just keep potassium inside the cell. If you open a potassium channel, you let positive charge out. That leaves net negative charge inside. If you open a chloride channel, you're allowing negative charge to come in. You're going to this inside of the cell is going to get further away from resting membrane potential. This is going to create an inhibitory postsynaptic potential, or IPSP. When you have IPSPs created, then it's going to take more stimulus to come in, more of these ligands to come into the channels that allow positive charge to come in to get you to threshold. So when you want to make a cell less likely to create an action potential, you create an IPSP in its plasma membranes inside this, in its cytoplasm on the postsynaptic side. Okay, so EPSP is getting you closer to threshold. IPSP taking you further from threshold. If you were at resting membrane potential, to get to threshold in this situation, you've got to have 20 millivolts of positive charge come in to go from minus 70 to minus 50. So you've got to get rid of, you have to cancel 20 millivolts of negative charge. You do that by bringing in 20 millivolts of positive charge. If you're down at minus 80, you now have to bring in 30 millivolts of positive charge to get to threshold. So you're less likely to have this postsynaptic cell create an action potential when it's going to take more positive charge just to get to threshold. With an EPSP, you're now at minus 60 millivolts. So the next stimulus that comes in only needs to make up 10 millivolts of positive charge. So that postsynaptic neuron is more likely to depolarize. Temporal summation or spatial summation, either one can occur in a postsynaptic neuron. We come back to this slide. This is our postsynaptic cell here in yellow. All of these are presynaptic neurons. If we stimulate this blue neuron repetitively, and by doing so, the neurotransmitter is continually released. It's in high concentration, close to the receptors on the postsynaptic membrane. So when one neurotransmitter is broken down by an enzyme or cycles off the receptor, another one is there to bind. You keep those channels open. Whatever they're doing, either letting positive charge in or creating negative charges inside, they're just doing that on a long-term basis. So with repetitive stimulation of one neuron terminal, you get what's called temporal summation. Temporal means over time, you are getting charges to add together. What essentially you're doing is keeping that channel open, so whatever charge is moving through that channel continues to move through that channel. The other way you can sum is by having different neurons, maybe green, blue, and red. Is that red? Can't tell. Brown? Well, red, blue, and green all fire at the same time. If they're all letting positive charge accumulate, then the positive charge adds together very quickly, you get to threshold. If one of those is inhibitory and two of those are excitatory, depends on the sum of all those parts when you reach the axon hillock. Is it enough to get to threshold or not? So when multiple neurons are going to open channels, then that is spatial summation. You all understand space. Each of you is in your own space. If you open channels in your space and all the charges then accumulated somewhere, you sum up those charges. And then you have spatial summation. Temporal, you have to remember what the word temporal means, and that is that over time something is occurring. Here, over time, the summation is the charges that are running through a particular channel. So here, we've got 3 nerve terminals. Nerve terminal 1 is excitatory, EX. Nerve terminal 2 is an EX, so excitatory. Nerve terminal 3 is IN, so inhibitory. We stimulate number 1 twice, but the stimuli didn't arrive very close together. So after you excite it once, it's back to resting membrane potential before the second stimulus arrives. All you end up having are 2 identical excitatory postsynaptic potentials, but you get no summation. In order to get summation, some of the positive charge from the first EPSP has to still be there when you open more channels with the second stimulus. And that's what's shown here. The arrows are indicating the time at which the stimulus arrived. Here, the time between the successive stimulus